All right, so now that we talked about prokaryotic cells, there are two domains that contain these prokaryotic cells. So the first one we're talking about, letter D in your outline, is the domain Archaea. So I want to just point out here that this is our three-domain system. This is the domain Eukarya. We're going to talk about that very soon. The domain Archaea that we're going to talk about, in the domain Bacteria. So both of these domains of prokaryotic cells. And you really don't need to worry about all the written stuff here. This is just a appreciation of the three domains. And we're going to talk about these two groups here, or I guess just extremophiles and methanogens. So the first sentence under domain Archaea is for you to be aware that the cells in this domain are generally found in places that we consider extreme. So if we have really extremely salty environments, you're going to find Archaea predominantly thriving in these environments. If you have super hot areas, right? So hot springs, these areas that are maybe too hot for eukaryotic life, you'll find cells from the domain Archaea here. So what we consider extreme, they thrive in these areas. So that's why we call them extremophiles. Except again, remember, the word extreme is from our point of view. It's not extreme for them. The suffix phile means to love, all right? So they love these habitats. It's not something that they tolerate or they have to put up with. They thrive and love these environments. So the word thermophile, introduced to you in your outline, is a cell that loves to thrive in hot environments. But I do mention that they're found elsewhere as well. They're found in sort of quote-unquote normal environments that we think of as sort of normal, or that can sustain life. Our second category of Archaea is methanogens. So methanogens are a group of cells that when they metabolize their organic molecules, they create methane as a waste product. So methane is a greenhouse gas. It's also used as a combustible energy source, right? So we can power things on methane. Methane catches on fire. So these are two images that I thought are pretty striking. The first one, of course, I made because cows do have methanogens in their guts. So you can see in the outline, they actually help the cattle and deer digest the grass that they need to eat. Grass is, predominantly cellulose. They can't digest it, so they have these methanogens help out. But as a sort of side effect, or I don't know if side effect's the right word, but as a result of having all these methanogens in their gut, they're creating a lot of methane in their intestines, and that methane gas has to escape somehow, right? So it's either through the two ends of their digestive tract, right? It's either through their mouth or it's through their anus. So methane has to get out. So I have a methane plume here because cows burp, or they have flatulence that can have methane in it. They contain methane. And so this is one of the reasons if you are a vegan or a vegetarian and you want to argue with somebody against, or like, why should we stop eating, let's say, beef, right, in this case, because it is a sort of greener diet to have. If you don't support the cattle industry, right, then you'll have less cows on the planet, and less cows mean less methane. And so methane is a greenhouse gas. It traps the heat on the planet. And so if you minimize the amount of cows around, then you can minimize the methane put out by cows. So that's a sort of green argument for not eating beef. The second picture I have here is really cool. It's frozen methane gas bubbling up from the bottom of this lake here. When it froze over, the methane gas also froze. So this is from a lake in Canada in the winter. And normally, this methane would just bubble to the surface and get liberated into the atmosphere, right? But in this case, because it's so cold, it froze, and you can really see the tremendous amount of methane bubbles that we have trapped in this. So the methanogens, those bacteria, or I should say Archaea, right? They're on this bottom of this ocean. They're decomposing the organic molecules down there, and they're releasing their methane gas. Really cool picture. All right, so I covered the two categories of Archaea. That's all you need to know. We have extremophiles and we have methanogens. And then our next topic is strictly domain bacteria. So this is probably what you guys think of when you think of bacteria, right? So we have, this is the bacteria that we come in contact with on a day-to-day basis, the bacteria that make us ill. This is all from the domain bacteria. So if you look at what I have, not much, but they're beneficial as well as pathogenic. So beneficial means that they are helpful in some way. So let's take a look at what I have down here. They're beneficial because they can recycle nutrients in the ecological cycles. So they can help break down things like rotting food, dead and decomposing plants. So they help to take those nutrients that are trapped in something that's dead and break it down so that it can return to the soil so other things can use it. So really important for that. Really important for symbiotic relationships for plants. A lot of bacteria can actually form colonies within the roots or near the roots of plants along with fungi, and they help the plants in different ways. We use bacteria to help create foods. This is a fun topic because bacteria are used to make a few different foods. We talked about yogurt, right? How bacteria is used to ferment the sugar in milk. It sours, it ferments it, it makes yogurt. We talked about this already, Swiss cheese, how the bacteria in the cheese, when it's developing, creates a gas that makes those holes in Swiss cheese. Yeah, so bacteria help to make different kinds of foods. We talked about sourdough bread as well, using bacteria. Number 3, they are necessary to for our immune system to develop properly. So it turns out that when they raised mice in sterile environments, the mice, their guts didn't form correctly, that they needed some kind of stimulation or some kind of external, contact with the bacteria to develop. And also in our digestive tract, some of the bacteria that live there produce vitamins for us. So we have this very symbiotic relationship with bacteria in our gut. Number 4, we'll talk about that in just a second. Well, let's go into it now. So number 4 is bioremediation, which is in your outline. So, I'm sorry, number 4 is biotechnology. But let's jump to bioremediation. So bioremediation is using bacteria to clean up sort of our toxic waste in the environment. So if we look at what's happening here, there is an oil spill, and this person is spraying fertilizer onto the oil spill. The fertilizer is to encourage the growth of bacteria, and the bacteria will grow and digest the oil, because if you think about what oil is, petroleum, it's a hydrocarbon, and that's an organic molecule. It's hydrogen and carbon. It's no different than a sugar, really. And so these bacteria can digest naturally this petroleum. So really good for us, right, that we have something that exists naturally, but also we can genetically engineer. There's labs working on genetic engineering cells that can break down products that we have put into the environment and realize that they're very bad for the environment. So we need to clean them up, clean it up. This is another example of bioremediation. This is basically a picture of your wastes that come into a waste treatment plant. So when you flush the toilet, all of that waste comes into your water treatment plants, and it's the very first step is basically bioremediation. So we're using prokaryotes and fungi both to break down those larger organic molecules into smaller components. And this is just showing you that they actually stir it up to give more oxygen into that waste, because these microbes need oxygen. They're aerobic. All right. Let's just go back and touch on the biotechnology and sources of enzymes. So a lot of our bacteria are actually used as little machines to make drugs, to make things like insulin for diabetics, and also different vaccines can be made with bacteria. So we can engineer the bacteria to make different things that we would want them to make. Also a useful source for enzymes. And then lastly, of course, bacteria make us sick. So the word pathogen is the word used for anything that can make you sick. And they can also make toxins, right? And toxins are bad for you. They make you sick as well. So the best defense against bacterial diseases are sanitation, antibiotics, and education. And actually, it should be education, sanitation, antibiotics. Antibiotics should go last, or it should be sanitation, education. But antibiotics should really go last. So you don't want to overuse your antibiotics because overuse can cause your bacteria to become resistant to the antibiotics. But that's what we have here. So, now that we're doing these online classes, COVID, the disease caused by the coronavirus, is a virus. So there's not a whole lot of drugs out there to treat viral infections. So when you see the word antibiotic, this really refers to bacteria. Antibiotics kill bacteria, not viruses. So taking an antibiotic when you have a viral infection like coronavirus or the flu is not going to help. Okay, at all. They're completely two different things. Apples and oranges. All right, so let's take a look at an example of a bacterial infection. This is Lyme's disease. Lyme's disease is on the rise in the United States. And they think that global warming has a role here. But Lyme's disease is caused by this bacteria. So working from the right, this is a spiral-shaped bacteria, right? So this is a spirochete. And the spirochetes are actually found in the guts and saliva of ticks. So ticks will carry the bacteria, and then the ticks will actually, when they bite you, right, this person shows this huge rash. When they bite you, they deposit the spirochete into your body and causes that Lyme's disease. One of the hallmark signs of Lyme's disease is this rash. It's a bullseye rash that rings out, but not always. So this is the key. Some people have Lyme's disease, but they never have that rash. So you can't count on seeing that rash for Lyme's disease. You can get Lyme's disease without it. But if you do get a rash, it looks like this. It's a bullseye rash. All right, but this is an example of a pathogenic bacteria, right? All right, so I think that's it. Yep.